The research we're presenting here is basically a summary of 7 years worth of effort to develop a needle-free, non-injectable vaccine against Ebola. I actually was contacted by one of my collaborators who was based at the National Microbiology Laboratory in Canada. I met him at a conference and he gave me his business card and said we needed to work together. And so when I got home, I actually looked and saw what he did, and he's a first responder to outbreaks in Ebola in Africa. And so I called him and I said, exactly what do you want me to do, with respect to what you do? And he was like, I've been to, places where this has been a problem, and needle vaccines are just not really, needle-based vaccines are really not going to be, the solution. So, I know you're a pharmacist and you can potentially fix this. We should write a proposal together to develop either something that could be administered nasally or that could be administered orally and get away from the needle-free or the needle-based vaccines that are a big issue with respect to Ebola in Africa. Well, there are no treatments on the market or vaccines against Ebola at this point. There are two vaccines that have been identified by the World Health Organization as potential candidates that are being currently either tested in humans in a phase 1 trial or ramping up for that. However, both of those vaccines are in the injectable platform right now. So again, ours is novel from the fact that it is a nasal vaccine in a single dose and long-lasting. So we've had discussions with the National Institutes of Health and Department of Defense, actually, but we were kind of in a holding pattern until our data had been scrutinized and reviewed by the scientific community and recently accepted. So Saturday, over the weekend, the publication describing these studies has been accepted for publication in a journal. So now we're at that crossroads. We have green lights ready to go, but we just need some funding sources. So we're at that point to see, again, where we can get resources and funding to further on our efforts to get this to the people that need it the most. We chose to present this work, as our first time in public at AAPS because we really believe that this is a unique opportunity for someone with skills in, pharmaceutical science and formulation sciences. And it was a unique opportunity, again, to be involved in something that had global impact. I had pharmacy students get involved in this, again, never in their entire career that as they were applying to pharmacy school they would be involved in such a world impact project, and graduate students as well. So again, it really does purvey a nice story about how pharmacy can really have a global impact.
Well, my role came in later for the second study, which was the long-term study, where I produced the vaccine itself. So it's a virus-based vaccine. And then after all the samples were taken, I also analyzed them. We looked at antibody response and how the virus sheds in the body, where we can find it afterwards. So that's the virus, the vaccine itself. So just kind of analyzing all the data afterwards. So our vaccine is another virus. It's called adenovirus, and you can take all parts of the adenovirus and add other parts. So what we did is that we took the glycoprotein of the Ebola virus and put it into the adenovirus. And that way, when you get the adenovirus intranasally, or either way you're getting it, you'll get a response to that glycoprotein from the Ebola. So we're not giving you Ebola, we're giving you another virus, which is really important because we don't want anyone to get sick from Ebola by accident. When I decided to go to graduate school, I knew I wanted to do immunology and I knew I wanted to do virology. So what I did a PubMed search, so you can search articles, and then I found Dr. Croyle through that and I read her articles. And the Ebola vaccine is something that could really make a difference in the world, so that's why I wanted to work on this project. And I was fortunate enough that she accepted me into her lab, and I feel like where we are right now, we could actually really help people, and that's kind of the purpose of this career, I feel like.